Hi guys! In this video, we will discuss Chapter 5, Population Genetics, Part 1, where the population is in equilibrium. These are the learning objectives that you need to achieve at the end of the lesson. At the end of the lesson, students should be able to explain population genetics, gene pool, allele frequencies, and genetic equilibrium. The students should also be able to state Hardy-Weinberg law, explain the 5 assumptions of Hardy-Weinberg law for genetic equilibrium, and calculate allele and genotype frequencies. Let's begin. Given that a population is large in size, consists of members that mate randomly, not subjected to natural selection, not experiencing mutation in their genetic information, and no migration, or in another word, movement of individuals into or out of the population is in equilibrium. Hence, the population is not evolving into another species. Hardy-Weinberg principle states that in a population that is in equilibrium, the genotypic and phenotypic frequencies remain the same or constant from generation to generation. The concept of frequencies, just like percentage or ratio, it refers to parts from a whole portion. What are the whole portion we're talking about? We can talk about the total alleles that is present in the population. The total number of dominant allele plus the total number of recessive allele of a particular gene in that population will give you the total allele of that gene in the population. We can also talk about the total individual that is present in the population. The total number of individual that is homozygous dominant plus the total number of individual that is heterozygous plus the total number of individuals that are homozygous recessive in the population will give you the total individual in the population. These two whole portions can connect to each other. We will learn this in detail later on in the video. However, just for a quick example, you can find the total allele for a gene in the population through total individual in the population. Since each individual in the population will carry 2 alleles for that gene, to find the total allele for that gene in the population, we just simply times the total individual in the population by 2. If you are required to find the frequency or proportion of the dominant allele in the population, which often designated by the letter P, you just simply divide the total number of dominant allele by the total allele of that gene in the population. If you are required to find the frequency or proportion of the recessive allele in the population, which often designated by the letter Q, you simply divide the total number of recessive allele by the total allele of that gene in the population. P and Q are known as allele frequencies. If you are required to find the frequency or proportion of homozygous dominant individual from the population, which often designated by P square, you simply divide the total number of homozygous dominant individual by the total number of individual in the population. If you are required to find the frequency or proportion of heterozygous individual in the population, which often designated as 2PQ, you simply divide the total number of heterozygous individual by the total number of individual in the population. If you are required to find the frequency or proportion of homozygous recessive individual in the population, which often designated by Q square, you simply divide the total number of homozygous recessive individual by the total number of individual in the population. P square, 2PQ, and Q square is known as genotypic frequencies. Now let's have a look at this example. There are 500 individuals in the population. There are 50 recessive alleles present in the population. Given that the population is in equilibrium, find the frequency of the recessive allele. We know that to find Q, or to find the frequency of recessive allele, we have to divide total number of recessive allele to the total allele in the population. From here, we know that there are 50 recessive alleles, but we are not given the total number of allele in the population. What we are given is that there are 500 individuals in the population. Since one individual carries 2 alleles for that gene, 500 individuals means that there are 1000 total allele in that population, giving you the value of Q as 0.05. From your answer, find the frequency of the dominant allele. We know that the total number of dominant allele plus the total number of recessive allele will give you the total allele for that gene in the population. Therefore, the frequency of the dominant allele plus the frequency of recessive allele will give you the whole portion. In other words, P plus Q equals to 1. From here, we can find the value of P. P is equal to 1 minus Q, so therefore, P is equal to 1 minus 0.05, so therefore the frequency of the dominant allele, or P, is equal 0.95. Now let's take a look at another example. In bears, having black coat is a dominant trait compared to having white coat. In a non-evolving population of 1000 bears, there are 750 black bears. Find the frequencies of white bears. We know that white bears must be homozygous recessive individuals. So therefore, frequency of homozygous recessive individual is equal to the number of homozygous recessive individual divided by total number of individual in the population. Since we know that having black coat is a dominant trait, and there are 750 bears with dominant trait, meaning there are 250 bears that are homozygous recessive, or in other words, having white coat, giving the frequency as 0.25. For your information, we can switch information between genotypic frequencies, p square, to pq, and q square, to allele frequencies, p and q. Take the above question for example. Find the frequency of black bears that is homozygous. To find the frequency of black bears that is homozygous, we cannot simply do p square equal to 750 divided by 1000. This is because black individuals can be homozygous dominant or heterozygous. So therefore, the 750 black bears consist of homozygous dominant individuals and also heterozygous individuals. We can use Q square, which is the frequency of homozygous recessive individual, to find Q, which is the frequency of recessive allele. Since we know that Q square is equal to 0.25, therefore Q is equal to the square root of 0.25, giving us the value of 0.5. Since we know that the frequency of dominant allele plus frequency of recessive allele will give us the whole portion, or the total number of alleles in the population, so P plus Q is equal to 1. P is equal to 1 minus Q. P is equal to 1 minus 0.5, so therefore P, or the frequency of the dominant allele, is 0.5. We still have not answered the question yet though, because the question is asking us to find the frequency of black bears that is homozygous. This means that the question is asking us to find p square. So therefore, the frequency of homozygous dominant individual, p square, is equal to 0.5 square, giving you the value of 0.25. The question then continued asking us to find the number of homozygous black individual. Since we already know the frequency of homozygous dominant individual, to find the number of homozygous dominant or homozygous black individual, we simply multiply the frequency to the total number of individual in the population, like this, 0.25 times 1000, giving us the value of 250 individuals. The question continued by asking us to find the frequency and number of heterozygotes in the population. Since we know that there are 750 black individuals, and from our previous calculation, we found out that there are 250 homozygous dominant individual, we can quickly calculate that there are 500 heterozygous individuals in the population. Or to put it neatly, we say number of heterozygous in the population is equal to total number of black individuals minus total homozygous dominant individual, giving us 500 individuals. To find the frequencies of heterozygotes in the population, or 2PQ, we simply divide the total number of heterozygotes by the total individual in the population, in this case 500 divided by 1000, giving us the value of 0.5. To check, when you add up all the genotypic frequencies, or in other words, frequency of homozygous dominant plus frequency of heterozygotes plus frequency of homozygous recessive, you will get the whole portion, or in other words, p square plus 2pq plus q square should give you the total of 1. If not, there must be something wrong in your calculation. Let's have a look at another example. State the Hardy-Weinberg principle. The Hardy-Weinberg principle states that in a population in equilibrium, the allele and genotypic frequencies remain the same from one generation to another generation. List 2 conditions for the principle to be achieved. There are 5 conditions. You can mention any 2. Large population size, no migration, no mutation, no natural selection, and random mating. In a randomly breeding population of mice, black coat is dominant to white coat. In the population, 36% have white coats. Calculate the genotype frequencies of black coat mice in this population. We know that black coat mice can either be homozygous dominant individuals or heterozygous individuals. So these are the 2 genotypic frequencies that we need to find: the frequency of homozygous dominant individual and also the frequency of heterozygous individuals. We are given that 36% of individuals have white coats, so this means that frequency of homozygous recessive individuals, or q square, is equal to 36 divided by 100. From here, we can find the frequency of homozygous recessive individuals where we square root q square, giving the value of 0.6. Since p plus q is equal to 1, the frequency of the dominant allele, p, is equal to 1 minus q, 1 minus 0.6, giving the value of 0.4. From here, we can find the frequency of homozygous dominant individuals, p square. We simply square 0.4, giving us the value of 0.16. From here, we can also find the frequency of heterozygous individuals, which is 2pq. We simply multiply 2 by 0.4 by 0.6, giving us the value of 0.48. To check the frequency of homozygous dominant individual, p square, plus the frequency of heterozygous individual, 2pq, plus the frequency of homozygous recessive individuals should give you the value 1. If you do not get the value 1, there must be something wrong with your calculation. The next question goes, in a human population, the frequency of recessive individuals for extra long eyelashes is 90 per 1000. What is the individual percentage of this population that carries recessive allele but displays the phenotype of short eyelashes? What are we looking for? We are actually looking for percentage of heterozygotes. Given the frequency of homozygous recessive individuals, q square, is 90 over 1000. From here, we can find the frequency of recessive allele, q, by finding the square root of q square. This gives us the value of 0.3. Since p plus q is equal to 1, the frequency of the dominant allele, p, is equal to 1 minus q. 1 minus 0.3, giving us the value of 0.7. From here, we can find the frequency of heterozygous individuals, 2pq, simply by multiplying 2 to 0.7 to 0.3, giving us the final value of 0.42. The question is asking us to find the answer in percentage form. So therefore, to find the percentage of heterozygous individuals, we simply multiply the frequency of heterozygous individuals 2pq by 100, giving us the value of 42%. The last example. On St. Helena Island, wingless insects established beside long-winged insects. Long-winged insects are carrying the dominant allele. In a population of 5,000 insects, 950 are long wings. By using the Hardy-Weinberg equation, calculate the frequency of wingless allele, or in other words, the frequency of recessive allele. Since we know that having long wing is a dominant trait, and there are 950 individuals with dominant trait, meaning the rest are individuals who are homozygous recessive individuals, or being wingless. From here, we can find the frequency of homozygous recessive individuals, Q square, which is 4050. From Q square, we can find Q, or the frequency of recessive allele, simply by finding the square root of Q square, giving us the value of 0.9. Since we have found Q, or the frequency of recessive allele, we can find P, which is the frequency of the dominant allele. In this case, long-winged allele. Since P plus Q is equal to 1, P, or the frequency of the dominant allele, is equal 1 minus Q, 1 minus 0.9, giving you the value of 0.1. The third question is asking you to find the number of heterozygous insects in the population. We can first find the frequency of heterozygous individuals in the population, 2pq, simply by multiplying 2 to 0.1 to 0.9, giving you the value of 0.18. To find the number of heterozygous individuals, we simply multiply the frequency to the total number of individuals in the population. In this case, 0.18 to 5,000, giving you the value of 900 individuals.